Good afternoon, everyone. Thank you for being here for the first journal club of the Italian Society of Cell and Gene Therapy. My name is Carmelo. I am a PhD student at the Ospedale San Raffaele and an early career member of the SITGEC. With me, there is also Marika.
Hello. Hello, everyone. I am Marika, and I am a postdoc in cell and gene therapy. I've been working at the Ospedale Pediatrico Bambino Gesù in Rome, and me too, I am a member of the ECR board of Società Italiana di Terapia Genica e Cellulare. As Carmelo mentioned before, we are going to start our journal club series. And so this is going to be the first episode of the series. So thank you very much for joining us today. The webinar will be 45 minutes. And at the end, we will have a short Q&A session of 50 minutes. This webinar will be recorded. So please, if you don't want to be featured in the recording, you can just switch off your camera. And you can type any questions using the chat. At the end of the talk, Dr. Anastasia Conti will share her email address, so you can easily reach out to her for any questions.
Today, for the first episode, we are honored to have Dr. Anastasia Conti. She's a project leader of the San Raffaele Institute of Cell and Gene Therapy. She earned her PhD in Public Health Science in 2015 from the University of Parma, pioneering new research lines focused on understanding the transcriptional and epigenetic regulation of transposable elements in acute myeloid leukemia, AML. In 2016, she joined as a postdoc the SR-TIGET Institute, where focused on research on the role of transposable elements in AML, response to chemotherapy-induced senescence, and on the impact of inflammation and senescence on genetically engineered HSPCs for the development of future HSPCs-based clinical applications. Now, as a project leader, she drives independent research lines on the role of transposable elements in AML, poor response to chemotherapy, and on the molecular basis of bone marrow failure in telomere biology disorders. Today, Dr. Conti will talk to us about her last work, titled Senescence and Inflammation are Unintended Adverse Consequences of CRISPR-Cas9 AAV6 mediated gene editing in hematopoietic stem cells. And now, Anastasia, we can start our talk, and thank you again to join us for this first episode.
Yeah, thank you very much for your kind introduction. First of all, can you see my presentation? Yes?
Yes.
Yes.
Okay, that's great, thanks. Okay, so thank you very much for the kind introduction, Carmelo and Marika. It's my pleasure to being here today and for the invitation to launch this new initiative by the Italian Society of Gene and Cell Therapy. So as you mentioned today, it's my pleasure to provide you kind of an overview about our last work with the aim of dissecting the role of cellular senescence and inflammation in the context of CRISPR-Cas9 mediated genetic engineering in hematopoietic stem and progenitor cells. So, but before directly going to the study, actually just let's do a step back. I don't know how familiar you are with the CRISPR-Cas9 tool, but just to wrap up a few important concepts that will help us in understanding better the work. CRISPR-Cas9 it's a new technologies which allow us to induce a double-strand break into the genome of the cells. And then if the cells repair via non-homologous end joining, that is an error flow mechanism, you can create a gene disruption, so insertion or deletion in the gene. While on the contrary, if the cells can employ and engage the homology-directed repair pathway, having a DNA donor template, they can insert it into the genome, and this can allow the restoration of the function of this gene. What we know in the context of homology-directed repair year after an HDR correction in hematopoietic stem cells is that it's a super constrained process because these cells are mainly quiescent, and so generally given that HDR occurs in SG2 phase of the cell cycle, these in the past years allow us the employment of pre-stimulation conditions in order to let the cells at least to cycle for a half. And so their exit from quiescence is extremely important to perform HDR in this cell type. So as you may know, genetic engineering is a site-specific gene correction strategy for a plethora of immunohematological diseases. HSPCs are extracted from the patient's ex vivo culture, and we perform genetic engineering using artificial nucleases able to induce a double-strand break into a predetermined genomic sequence. Then, in the context of HDR, in hematopoietic stem cells, we use as a carrier of the DNA donor template an adeno-associated viral vector serotype 6. These allow us to insert precisely in the targeted site the corrective sequence, and then the gene is restored and its expression is under its endogenous promoter. The corrected cells are finally infused back into the patient, where they will give rise to a progeny of all corrected differentiated cells. However, when we started this project, we thought that despite the rapid development of genetic engineering tools and the recently reported clinical success, a few challenges remain to be faced, such as from the basic biology standpoint, the limited understanding of how hematopoietic stem cells respond to ex vivo genetic engineering. From an efficacy standpoint, the low yield of HDR-corrected HSPCs and their delayed long-term oligoclonal reconstitution, and finally, the increased risk of on- and off-target genotoxicity. So in this context, we reasoned that there was an urgent need to gain mechanistic insight into how hematopoietic stem cells respond to the genetic engineering procedure in order to advance gene and cell therapies. So our first questions were how hematopoietic stem cells respond to genetic engineering and how we could counteract the detrimental effect induced by the procedure. In our first study, in collaboration with our director in the Institute, Professor Naldini, we perform genetic engineering not only using CRISPR-Cas9, but at that time also we used zinc finger nucleases. And what we found was that when the cells experience a few double-strand break, there was just a modest p53 activation. On the contrary, when performing multiple double-strand break into the cells, there was a massive activation of the p53 pathway, strongly affecting the functionality of the cells. But what about the response of the cells to the full genetic engineering procedure involving, again, double-strand break induction plus DNA donor template delivered via an AAV6? Just to make a long story short, we found that the concomitant exposure to CRISPR-Cas9 and the AAV6 vector in hematopoietic stem cells at the single cell level strongly activated p53 pathway, impairing the functionality of the cells after transplantation into immunodeficient mice. In that context, we envisaged a strategy based on p53 temporal inhibition via the mRNA called GC56, which allows us to have an higher engraftment of the cells. As you can appreciate here, when looking at the red curve, this is the engraftment of the cells pre-treated with p53 inhibition at the time of genetic engineering over the low engraftment of edited cells without any treatment in blue. And moreover, p53 inhibition allowed also to have a more polygoclonal graft. That was the first example on how temporary manipulation of DNA damage response pathway could affect the functionality of the cells. So we reasoned that DNA damage response cascade and associated pathway could have an impact in the functionality of the dynamics of repopulation of hematopoietic stem cells post-genetic engineering. However, given that p53 inhibition might aggravate the genotoxicity risk of the genetic engineering procedure, we thought that we could act downstream. And we came up with the context in this new story that we are discussing today that the persistent DNA damage response activation leads to the establishment of cellular senescence. I don't know how much familiar are you with the senescence, but actually senescent cells are cells that, despite being still alive, are further unable to proliferate. Indeed, they activate the cell cycle inhibitors P16 and P21. They have the accumulation of the lysosomal enzyme called senescence-associated beta-galactosidase. They have extrusion of chromatin, so the so-called micronuclei, and then they have morphological and metabolic changes, as well as widespread activation of inflammation. And these cytokines can also secrete in the surrounding microenvironment, affecting overall the whole cell population, just cells that have not experienced editing. So the reason that the gene editing procedure, which involves prolonged time in culture, exposure to high vector doses, and double-strand break induction, may have an impact in the detrimental functionality and hyperfunctionality better of these cells after genetic engineering. To address that, we perform a very simple experiment. We used both cord blood and mobilized peripheral blood-derived CD34 hematopoietic stem cells, and then, just in order to have at least half of them cycling, as we said before, we pre-stimulated them for 3 days in our early active cytokine and stem preserving medium. Then, after these 3 days of pre-stimulation, we perform either CRISPR-Cas9 editing against a safe harbor locus in HSPCs, AVS1, or the clinically relevant locus IL-2 receptor gamma mutated in the SCIDEX1 immunodeficiency. Then, given that we want to have HDR, we provided DNA donor template via AAV6, and in this case, in the cassette, we had also a GFP to track the green cells, so the HDR edited cells over time, or a single-strand oligodeoxynucleotide. As negative controlled, we use cells that just experienced the sole electroporation, hereafter named MOCL. And we collect these cells early and late post-genetic engineering for further cellular and molecular analysis. First, we measure DNA damage response activation. In terms, here I reported the P21. As you can see, respect to cells having just electroporation here, or cells having just double-strand break induction with a nice specificity guide RNA, or again, single-strand oligodeoxynucleotide used as a template for HDR, there was a massive upregulation of P21, just in the condition in which cells have experienced HDR using an AV6. And this upregulation was also in the context of HSAV6 treated cells. Broadly speaking, at the transcriptional level, was also associated with not only p53 pathway activation, as we previously reported, but also as an increased expression of inflammatory-related categories, as well as senescence-related categories. And this was not only true at day 1, our conventional time point when we perform transplantation into immunodeficient mice, but also later on. And this increase at the transcriptional level in senescence categories was also coupled with an increase in the accumulation of senescence markers, such as, for example, the senescence-associated beta-galactosidase are reported here, in which you can clearly see that again, there is the higher accumulation of this marker in the 2 blue conditions, so when cells experience HDR using an AV6 versus MOCL cells or HS, single-strand oligodeoxynucleotide ones. At that stage, we wonder if the vector could have an impact in this detrimental functionality of the cells. And to check that, we perform an experiment employing increasing doses of the AV6 vector. And as you can appreciate here, we found a progressive impairment on the functionality of the cells, so in their ability to generate colonies in CFU assay, increasing the vector dose versus control. And interestingly, when we check at the senescence accumulation here in terms again of senescence beta-galactosidase in the progeny of these cells, you can clearly see, if you follow here the green, that there was an accumulation of senescence beta-galactosidase, especially in the HDR progeny of HSAV6 edited cells. And this was not just true for senescence beta-gal, but also for P16 marker. So there was an activation of senescence in the progeny of edited cells. We wonder if that could be the case also in vivo. So we transplanted the cells and we wait 18 weeks. And just to make a long story short, we found exactly the very same we found in vitro, also in vivo. Indeed, here again, if you consider the CD45 positive cells, so the overall engrafted human cells. Again, you can see that there was the higher accumulation of senescence beta-galactosidase, especially in cells having GFP, so cells repairing by HDR. And this was true comparing this fraction of cells to the not HDR edited ones, here GFP minus, as well as the controls. So cells AAV6 means cells just having experienced the transduction with the vector at the 3 different doses tested, as well as the mock-L cells. And this was just not only true in all the graft, but also in the different lineages. As you can appreciate here for the B cells, the myeloid cells, as well as the most primitive CD34 positive cells. So this meaning that cells having repair by HDR activate senescence in vitro, but after transplantation and after waiting 5 months into the mice, this higher accumulation of senescence markers is also found in the cells retrieved from the marrow. At that point, we wonder if this induction of senescence was due to the exogenous expression of GFP, or actually it was strongly associated with the HDR process per se. And we made a very simple experiment in which we compared side by side cells having experienced HDR using a cassette having a GFP as reporter, as well as another one having the more clinically relevant and physiologically delta NGFR. And as you can see here, by employing the 2 different cassettes, actually the cells had the very same levels of engraftment in the marrow after 18 weeks post-transplantation. But interestingly, when we checked at the senescence burden, we found again that there was the higher accumulation of the senescence marker, beta-gal and other that we tested in the study, especially in cells having experienced HDR. So both the GFP plus over the GFP minus fraction, as well as the delta NGFR plus versus the negative fraction. So it was HDR per se inducing senescence. At that point, we thought how we could mitigate this detrimental senescence and inflammation induction in edited cells, because I want to remind you that this study have also, let's say, in mind that we would like to apply these basic biology results to the clinical practice. So we thought that, of course, we could employ p53 inhibition that we used in our previous story to prove that there was an higher engraftment of the cells and a polyclonal graft composition, so as kind of a positive control. But again, given that we want to also explore downstream pathways with respect to p53, we decided to inhibit an FA-kappa beta inflammatory pathway, as well as IL-1 signaling pathway. Why IL-1? Because IL-1 is strongly associated with hematopoietic stem cells dysfunction, DNA damage, and also senescence. We inhibit IL-1 signaling pathway using Anakinra, an already FDA-approved drug for rheumatoid arthritis and other inflammatory-related disease, while we use this compound SC514 to inhibit an FA-kappa beta. So just to make a long story short, because of course in the paper you can find also the characterization using these compounds in vitro. When we moved in vivo, we found that, as you can see here, when looking at the engraftment of the human CD45 positive cells over time in peripheral blood, we had a stable higher engraftment when cells were edited in combination with p53 inhibition, IL-1 inhibition with Anakinra, or the combination of the 2 inhibitions over cells having experienced just gene editing here in blue. And this higher engraftment was not only true over time post-transplantation up to 18 weeks, but also in the different lineages after at the end point. So I mean hematopoietic stem cells, B cells, and myeloid ones. And this higher engraftment was associated with a decrease in the accumulation in these cells of senescence markers. Indeed, now, if you again follow the green, respect to cells having just experienced gene editing, we had a trend of decrease while treating the cells plus inhibitions up to 18 weeks post-primary transplantation. And this was also even more exacerbated, so this dampening in senescence marker accumulation was even higher when we performed secondary transplantation, suggesting that even if the cells experienced the inhibition just during the gene editing procedure, if we transplanted them one day after gene editing, they continue to maintain kind of a memory of their past treatment. And the higher engraftment they have is strongly connected with this decrease in senescence activation, even after secondary transplantation. Then, in collaboration with Professor Naldini in the Institute, we also employed a barcoding strategy in order to keep track of the cells, so the clones that were able, the HDR-edited clones that were able to engraft. And as you can see here by bar-seq, we found that when we retrieved from the mice cells having experienced before transplantation p53 inhibition, we had this polyclonal graft composition as we previously published. But interestingly, and this is the novelty in this story, we also found polyclonal graft composition upon IL-1 inhibition. And this was not accompanied with an increase in clones repair in viral homologous enjoining. So we were actually acting mainly in increasing the polyclonal reconstitution of individual HDR-edited HSPCs. At that point, as I said, in the perspective of applying this to the clinical translation, so the clinical practice, we thought it was worth to also check genotoxicity risk. We employed different assays, both ex vivo and in vivo. And let's start with Cas-seq. In collaboration with Professor Katomen in Germany, indeed, we performed this assay, which allows us to check all the translocation events and off-target sites genome-wide. And as you can see, we didn't find any difference and any increase of translocation events or off-target ones across treatments, while compare these with cells having experienced double-strand break induction, nor on-target aberration. So the percentage of on-target aberration was the very same across all conditions. Of course, in all conditions, it was higher respect to MOCL, but no differences again, while treating the edited cells with different kind of inhibitions. Then we decided also to explore a bit more what about micronuclei formation. Why? Because we said, okay, David Pellman's group a few years ago explained the phenomenon called chromotrypsis. So this extrusion of chromatin from the nucleus. Given that, as I explained you before, micronuclei are also strongly associated as a feature of cellular senescence, we thought that this could be an indication of having senescence, and also that this could be used as a new genotoxicity assay. So just to make a long story short, what we found was that respect to all these negative treatments, so cells again having experienced electroporation or double-strand break induction or the transduction with an AAV6 alone, the cells having experienced the full genetic engineering procedure with an AAV6 had a higher percentage of micronuclei. And this was true not just for mobilized peripheral blood HSPCs, but also in the context of cord blood-derived HSPCs. When performing p53 inhibition, we didn't have any increase in the percentage of micronuclei positive cells. But interestingly, as you can see here, when we perform IL-1 inhibition as well as an FK beta inhibition, we found less micronuclei, suggesting that this reduction in and transient manipulation of inflammatory-related pathways reduce one of the key assays of genotoxicity. Then we decided to move forward, and in collaboration with Stephanie Xi in Canada, we also performed an analysis of large deletions downstream the targeted site in single-peaked colonies. Here is the overview of what we found in mobilized peripheral blood-derived cells, colony-derived cells. And what you can see here is that in the context of gene editing, there was an increase in large deletion respect to Mockel cells that was maintained at similar level when performing p53 inhibition, but again, when performing IL-1 or NF-kappa beta inhibition, especially in the context of mobilized peripheral blood-derived colonies, there was a significant decrease in the occurrence of large deletion in this clonogenic output. That was maintained as a trend also in the context of cord blood colony-derived cells. Finally, and I think this is the most interesting data we found, when looking at what happened in the bone marrow-derived CD34 positive cells post-transplantation via whole exome sequencing, we reported just a trend of increase, a slight increase in the mutational rate in these cells when considering the editing plus p53 inhibition over just edited cells, while interestingly a significant decrease, I would say, in the mutational rate when cells were treated with anakinra. So this means that the cells that again just saw anakinra at the time of gene editing for 24-hour ex vivo, after transplantation, 18 weeks post-transplantation, had less senescence activation, less genotoxic readouts, also less mutational rate. And when instead looking at the percentage of variant types, across conditions, we didn't find any difference in terms of single nucleotide variants, deletion or insertion, nor differences in the percentage of variant impact that we found. So I mean, mutation having an high, moderate, or low impact. So just to conclude, I hope I have convinced that by integrating transcriptomic and cellular analysis, we found a widespread activation of DNA damage, of inflammation and cellular senescence in the context of CRISPR-Cas AAV6 mediated gene editing, and that this senescence in the human graft long-term affects cells mainly that have undergone HDR, could be mitigated not only by transient p53 inhibition, but also with anti-inflammatory strategies such as anakinra we employed, and contributes to the oligoclonal reconstitution of the cells and their impaired functionality post-transplantation. Of course, after checking genotoxicity, we were happy to see that there were no signs of genotoxicity while performing transient inhibition of these anti-inflammatory pathways or while performing transient senescence inhibition. So with this, I conclude thanking all the people involved in this study, Raffaella Di Micco, my supervisor, all the people working with me in the project, especially Katie and Federico, of course, all the lab members, Professor Naldini's group, our internal and external collaborators, all the funding agencies, and of course, all of you for your attention, and I will be happy to take any question. Thank you. Don't be shy.
Thank you, Anastasia, for this beautiful presentation. Maybe I can start. So you basically use the nuclease, let's say Cas9 and also Tinfinger, if I remember correctly. Do you think that the senescence output can change using different editing tools such as base or prime editing, since they are based on nicases, or also like dead Cas epigenome editing, so without nicking? Do you think that the output can change in your analysis?
Yes, thanks for the question. This is a good point. Yeah, I think so. And actually, we have data also suggesting that. Of course, experiments with prime editing are still ongoing, but I can tell you some preliminary data we got using base editing. Of course, in this context, the percentage of inflammation and senescence is lower respect to what happened when you employ the HDR. So when you have double-strand break plus especially AV6 transduction. But again, it's higher respect to Mockel cells. So this means that if you have just a very few percentage of cells having this slight induction of senescence, just performing electroporation, When you use base editing, this fraction is increased, but not at the very high level we measure when performing double-strand break plus transduction. And for the dead Cas9 you mentioned, and also even for the prime editing, even if we don't have any data up to now that is conclusive, I'm expecting to have less senescence respect to the context of double-strand break plus AV6, that I think is the most detrimental case, actually, that the cells can experience.
Thank you.
Presentation, very clear.
Hi, Lina.
Just a curiosity. I was
Yeah.
wondering, how do you think that anakinra can reduce the mutational burden? Just a curiosity, like
Yeah.
how inhibiting IL-1 can also have this effect. I mean, which is good.
Yeah. Thank you for the question. Yes, we actually didn't explore this too much, but we had some option, of course. First, we thought that because there are also other studies reporting that IL-1 signaling pathway is involved in some kind of mutation, that maybe using anakinra, we are preventing the induction of mutation. We are reducing DNA damage, or we are helping the cells to be better in repairing the damage. And so if they are better in repairing the damage, of course, this can decrease the mutational rate. Thank you.
Thank you, Anastasia, for
Thank
the talk. It was stunning, your work. So I'm really impressed. So just a curiosity because you mentioned the transcriptional profiling of your cells. Can you explain a little bit what you performed, especially in terms of single cell transcriptomics or bulk RNA-seq? I don't know. Yeah, thank you.
Yeah, sure. In the data I mentioned now, it was a bulk RNA-seq. So we just check what happened in terms of overall picture at the transcriptomic level. So this was a conventional bulk RNA-seq. But in the story, in the full paper, I mean, we also try to overimpose in our single cell RNA-seq in the previous story, so the p53 inhibition one, what could happen if we check in terms of senescence in the different subsets, this eventual expression or not of the markers. And what we found, and this is nice because this precisely correlates with what happened at the cellular level, that again, especially in most of the, let's say, populations, but mainly in the primitive HSC, we had a higher expression of senescence markers, or senescence categories better, in the fraction of GFP positive cells, that also in that single cell RNA-seq was the fraction of HDR additive cells. So this means that overall, if you check the transcriptional level in single subsets or in bulk RNA-seq or different cellular assay, in all cases, you have an induction of senescence and inflammation, especially in cells having experienced HDR. And I think actually this makes sense because we are pushing the cells out of quiescence. So we are just with the pre-stimulation time per se inducing a stress. Then when cells cycle, they are more prone to make error or actually to have a kind of a bigger detrimental effects. And the cells having HDR are actually the most cycling ones. So yeah, that's pretty much expected. Thank you.
Thank you.
Thanks to you.
Any questions from the audience? Don't be shy, please.
Yeah, just if you have curiosities, I don't know. It's a kind of an informal chat now. I know it's Friday afternoon, not maybe best timing to discuss science, but It's always a good time to discuss science, no?
Hi. Thank you very much.
Bravo.
The talk was really, really good.
Thanks.
I'm not an immunology person. I come from neuromuscular diseases, but I was kind of interested by most of the graphs that you showed us, mostly in the edited treated cells. They showed kind of a bimodal distribution of results, quite neat. So I don't know, I just wanted to ask you if you have something more to comment about that, or I don't know if it's something that you also found interesting.
Yeah, thank you for the question. Yes, you're right. Especially in the in vivo experiments, we had this kind of bimodal distribution, but this is just a matter of also the editing efficiency, no? So when we had higher editing efficiency, these cells were less able to engraft, and that's why the group with lower engraftment. Instead, for any reason that can span from the AAV you use, so the production or the efficiency of the nuclease, so on and so forth. When we had a very, let's say, smaller percentage of editing, I don't know, in the range of, let's say, 10%, these cells were a little bit better, and so they engraft a little bit more. So that's why you can see these two populations. But also when we perform statistics, it was nice because, of course, you have all the different experimental conditions at the same, let's say, mice, no? So in other words, in all the different conditions, you have the same cells, the one with low editing, the one with high editing, and it was nice to correlate that within the group, at least we could see or higher or lower engraftment, and the same for senescence. So it was kind of controlled, in other words.
Okay, great. Thank you very much.
Thank you for the question. I know, Carmelo, you have another question.
I'm not so, I mean, I don't know actually all the experimental procedure to do editing in HSPCs, but you showed the pre-stimulation, if maybe 3 days of pre-stimulation condition. Do you think that that kind of pre-stimulation can boost or in some way activate the senescence that you showed to us? And if you try to, let's say, shortening the days of pre-stimulation.
Yeah, thank you for this question. Yes, of course, as I said before, also discussing with Marika about transcriptomics, when you pre-stimulate the cells for 3 days, and so you force them to exit from quiescence, this can have an impact on the subsequent senescence induction you see. We tried to shorten the time window of this pre-stimulation in another story in the lab. But of course, I think it's intuitive that reducing the time in culture also reduce the percentage of HDR that we got. So in that context, we tried to, let's say, improve the condition. So maintaining them for 3 days, but inhibiting P38 in that context. So at least we had good editing, so good HDR percentages, and less inflammation, because in that context we didn't check senescence. Of course, you can always try to shorten the pre-stimulation time, but especially in a context of HDR, you are risking to have too few cells. So if you are lucky and your corrected cells is in the context of a disease in which just one single corrected cells can have a selective advantage, you can also think about shortening the time in culture. But on the contrary, if you have diseases in which you don't have a selective advantage, you need to maintain a good fraction of HDR. So in that case, you cannot shorten the time in culture. Does it make sense?
Thank you.
Yeah.
Thanks.
So if there are no more questions, Anastasia, thank you again to join us for this first episode of the SITGEC Journal Club.
My pleasure.
I remind you that in the chat you have the email of Dr. Conti. So if you have any curiosity, questions, you can also send an email to Anastasia to talk about science and to talk about this paper and the things that she talked to us. So we can close the session. Thank you again, Anastasia, for joining.
Thank you very much. Thank you very much. It was my pleasure, and I will be happy to discuss with all of you. Please just send me an email and we'll do it. Thank you. Bye. Have a nice
Thank
weekend.
you.
Thank
Bye-bye.
you. Thank you.
Thank you. Bye bye.
Bye-bye.
Thank